We may or may not have time at the end for questions, but again, we'll just see how it goes. Great. Thank you.
Thanks. Sounds like it's on? Yeah. All right. Hi, everybody. So thank you all for coming. I'd like to point out that I am not David Beasley. His talk is in 517D. Raymond Hedinger, I'm also not him. And I'm also not Augie Fackler, and they're all really good speakers. So you should really reconsider your choice to attend this talk. It will be recorded and available later. I'm going to talk today about how to interpret your own genome. I'm not going to tell you how to get your own genome, because it turns out that there is no commercial service that will just give it to you, and I'll talk about that a little bit at the end. But I'm going to try and introduce all of the problems and associated opportunities in genomics, and human genomics and genetics. And I like to bill these talks as my ongoing attempts to explain what it is I actually do as a professor to Terry Peppers, who's sitting over there, who about 4 years ago, when I started getting more and more into bioinformatics, said, I don't understand how Python and biology intersect, basically. And we'll see if this is any more successful than my last talk, which was so unsuccessful that he didn't even attend PyCon. I guess I should introduce myself a little bit. I'm now a professor at UC Davis in the School of Veterinary Medicine, which I'll talk about a little bit later. I've been at MSU, Michigan State University, for the last 5 or 6 years. I'm very into open science and open source and Python and genomics and other things. And some of those will be on display today during my talk. So please feel free to email me or ping me on Twitter or check out my blog if you're interested in some of the links associated with this talk. So I'm going to really try to avoid being a professor, which is challenging. But I feel like I have to introduce DNA, because that's what's in your genome. So you've all heard, hopefully, that the primary DNA sequence consists of A, C, G, and T. Most human cells contain approximately 6 billion of these A, C, Gs, and Ts in a particular sequence, in a particular order. They're divided into 23 chromosome pairs. Every cell, mostly, has 23 chromosome pairs. And the really, really important bit is these 6 billion base pairs of DNA, these 6 billion characters of DNA, and these chromosomes are the primary unit of heredity. And what I mean by that is that the main reason that we look like we do as humans and not like other animals, and that we look like our parents, is due to the content of these 23 chromosomes and these 6 billion bases of DNA. There are a lot of modifiers, and there's a lot of additional information, but my guesstimate would be that it's on the order of 5 to 10%, and I'm happy to talk more about that later. But this is sort of a 90% true statement, which is very common in biology. DNA is ridiculously complicated, especially in the way it's interpreted. And again, I'm going to try to avoid the professorial sort of delve into details that nobody really cares about. But basically, you start at the DNA level, and then parts of the DNA are turned into RNA, and then parts of the RNA are turned into protein. And some of this happens inside the nucleus of your cells, and some of this happens outside the nucleus of your cells. And there's lots of exceptions to all of this. And it's basically an incredibly complicated concurrent execution system where up to 20 or 30,000 loci in the genome are being turned into RNA. All of those are being turned into proteins at some rate, each in a different cell in a different way. And they combine and collaborate to lead from a single fertilized egg into us over a period of, well, depending on how you count it, let's say 15 years or so. And then there's a lot of complicated things that go on before and after that, and I'm not really going to go into that. The really important bit for this talk is to think about how inheritance and the generation of variation in this genome works, right? We all have different genomes to some extent. Even twins have different genomes by the time they've developed. And the way it basically works is that John here gets one genome from their grandmother, one genome from his mother, one genome from his father. Each of those then get recombined between the colors for John's own children, so that the grandchildren are a mixture of grandma and grandpa's genome on the father's side, and the same on the mother's side. And this recombination is one of the 2 ways in which a lot of genetic variation gets generated. The other is that there are about 300 to 600 mutations per generation. We don't actually know the exact number on average. I have a link that will be put in the blog post as to how this number is generated. And this is novel mutations per generation, that is, from a person to their germ cell that gets passed on to the next generation. So there's variation that comes from taking mixtures of genes and DNA from parents, and then there's de novo mutations that happen from scratch. So I think if there's one message you take away from this talk, it's that a genome sequence doesn't really tell you all that much in some sense, because we actually know very little about how biology works, which is one of the reasons why I do what I do, which is research genomics and try and understand how genomes connect to the underlying biology. So if we knew someone's genome sequence perfectly, which is actually also quite tough, I'm going to show you how to get an approximate estimate of what a person's genome sequence looks like. We could do things like say, well, this person has this disease and this person doesn't have this disease, and there are these things in the genome that correlate with that. We could say, well, your parents are probably these people and not these people. Actually, that's probably what it's most used for. We can probably identify ethnic origin, and I'll talk a little bit about that later. And then we could find known mistakes or problems in the genome, like, oh, an A in this position is known to cause this disease unless there are other compensating factors, and I'll show you a specific example of that. But it turns out that even if you completely ignore environmental factors, we actually know very, very little about how the genome turns into a person. So the genome is not the person. Even in the case of environmental factors, we don't know how to interpret it. And here's a little bit of background for why I gave this talk. So if you attended David Beasley's talk last year, I don't know how many of you did, but it was this great talk about how David got locked into a room every day by a bunch of lawyers and handed a really large stack of CDs with source code on them. And he had to interpret the history and the technical content of the source code on these CDs for the purposes of discovery and for the purposes of sort of a patent litigation. And the only problem was he was locked in this room. He had no internet connection. All he had was a Windows 98 machine with Python installed. So he loaded all the DVDs, all the CDs onto the computer and spent, it sounded like a pretty miserable couple of months of his life, very well compensated, but a couple months of his life, trying to figure out what all of this source code was, what the versions were, how they related to each other, when ideas had been introduced, on what CD. And then he would print things out because he couldn't actually take electronic media in or out of the office. And then he would bring that to the lawyers and ask for more information from the various sides in the litigation. So when I heard that talk, I thought, I really like the image, by the way, of feral lawyers roaming around outside. I married into a family of lawyers, so this is being recorded. I always forget. OK, so when I listened to David talk about this, I said, this is exactly what scientists do. Right? What they do is they take a massive amount of data. They're given what genomic scientists do. They're given a massive amount of data. They're told to interpret. They want to figure out what it all means. And there's a couple differences. One is you do have access to the internet and a worldwide collection of all of the different scientists out there. And you have access to some of their data and some of their conclusions. It depends on sort of the open science, open access, open data issue. There are tremendously important bits of information in this set of DNA. You want to know what it means because the meaning really holds the key, literally, to life and death in many situations. But for all intents and purposes, you have no idea how to interpret this DNA. And so I liked the analogy with Pearl 8, because Pearl 6 is already looking like it's going to be a massive mess, right? Pearl 8 surely is not going to be any better. But we're not going to have neither a specification nor an actual implementation of anything that looks like Pearl 8 for quite some time yet. And this is really where we are when we're looking at the genome. Lots of code, really no ability to understand what it does or how to run it or how to simulate it. And I think I omitted this comment from here just by mistake, but it turns out to actually be illegal to test drive human genomes. Right? You can't actually edit a genome and put it in an egg and see what kind of person it grows up into. Something to do with unethical and we can do that in model organisms, which is actually what a lot of biology is done in, like mice. Okay, but all of this having been said, even though we understand very little about how to interpret genomes, they're still incredibly useful. And the way that we've dealt with it so far, and we've only had a full human genome sequence for about 15 years, is we spent a lot of money sequencing one or a few genomes and then use that as a reference for interpreting all of the other genomes that we get. So the initial cost of the first human genome was estimated to be about $2.7 billion in 1991 dollars, which is, I think, probably more now. I don't know, $4 billion. The current human genome reference that's publicly available that you can go download using the instructions in the pipeline that I posted on my blog, it's from 13 anonymous volunteers in Buffalo, New York. Now, there's a couple interesting things about that. One is that the genome that we use to interpret everybody else's genome is probably mostly from white guys. And there's two reasons for that. One is white guys probably are predominantly who are sequenced in Buffalo, New York. And the other is that because of the sex chromosome, XX and XY, you tend to want to sequence an X and a Y, so you tend to sequence guys. Because you'll get one of their mom's X's either, you get their mom's X's either way. So the old technology that was used prior to having the human genome was to identify some interesting points of variation based on various research and then target those for further investigation. The current technology, and what I'm going to talk about today, is to sequence everything, sequence the whole genome, and then resequence the whole genome, and then analyze it in the context of the reference genome that we have. And the new technology coming down the pipe now and over the next 5 years is going to be to do more sequencing and do better sequencing to get a better idea of what's out there, to the point where we'll essentially be doing this $2.7 billion genome to the same level of quality for about $1,000 in a couple of years. So working with this kind of sequencing, so I'm going to be talking about short-read sequencing, and I'll introduce exactly what that looks like in a sec. But the overall overview of the process we have to go through to interpret things is you sequence, you generate some sequence, you take that generated sequence and you map it to your reference genome. You then ask where that sequence differs from the reference genome. Remember, we're not interested necessarily in how similar these people look to our reference genomes. We're interested in where there are points of difference, because that's going to highlight where there might be interesting disease variants or differences in heredity. We call these variants, these differences, and then we interpret that in light of what we know, in light of the databases that we have in front of us. So if you want to sequence a genome, you need about 250 nanograms of DNA at 2 nanograms per microliter, and then you send that to a company or a sequencing core, and for about $1,000, you generate a lot of data. And this $1,000 is what they actually charge you. There is some upfront investment required. The machines involved in doing the sequencing cost several $100,000. The $1,000 includes sort of maintenance cost, depreciation, and the cost of reagents to go from DNA to digitized, biochemical DNA to digitized DNA sequence. And so there's probably a couple hundred places around the world now that have the capability of doing this. And the trick is, as I'll point out at the end, the trick is figuring out how to talk to one to get them to agree to do your DNA for you, if you're interested in this. When you take your DNA and you feed it into this machine that does the sequencing or the digitalization, you end up with a lot of raw data. You basically end up with about 2 billion records that look like this: A, C's, T's, and G's, followed by a random formatting character and preceded by a random name that contains very little information. And then a quality score which tells you how likely each one of these bases is to be wrong. Essentially what you're doing is you're taking this genome and you're observing it in a particular way. You're measuring it in a particular way. And there's measurement error associated with that observation. And these characters here are numerical characters that tell you how likely it is that observation is to have resulted in the wrong character. So then you take all of these short sequences of DNA. They tend to be in the 150 base pair range currently, so 150 characters. You map them to your reference genome. They come out of the sequencer in a format that's called FASTQ, which is what I showed you before. You map them to the Buffalo reference genome, and then you get out a file format called BAM. And I'll show you an example of that. And what you're really getting out at the end is something like this, where you have your reference, all 3 billion characters. I could only fit about 100 on here. And then you have a bunch of places where all of these lines are these individual measurements that you got of the genome. And then the differences are what you see in characters here. So if you see a C here, that means that up here there was I can't actually see either a T or a G. And in this measurement of the genome in this particular location, there was a C in one of the 50 or 100 measurements that you took. You then take all of these lined up observations and you say, are there any columns where everything agrees that there's a difference from this, right? And you sort of have to deal with the fact that there's noise because you have measurement error, and then but you also have real signal, which is what's actually different in what you're sequencing from the reference genome. And to give you an example of what that looks like, here you can see that approximately half of the data that you're generating contain a gap of 4 bases in this particular location. And the rest, this all looks like random. The rest of this looks like random measurement stuff, but this is actually a situation where one half of each chromosome, of the chromosome pair for this, has a difference from the reference genome. At this point, you're now in both a good position and a bad position. You've done a lot of work. The estimate for this is about 1,500 CPU hours to get to this point. And now you have what are called these variants. It says at this position in the reference genome, the sample you gave me differs in such and such a way. And now you have to go, well, I wonder what these variants are. Well, if it's a variant that's known to have an effect, and again, I'll show you a specific example of that in a bit, you're golden. You say, OK, well, this variant is probably linked to this disease. If it's in a gene, you have some hope. But only about 2 to 3% of the human genome actually codes for genes. So very few of your variants are actually going to be linked to something where you have some idea of what the function is. If it's in a gene and it does have some obvious effect, for example, it truncates the gene and it modifies the gene sequence in such a way as to break the gene, then you may have an obvious effect out of that. Or if it's been associated with some effect like, hey, we don't know if this is the causal variant, if this actually is what breaks things, but it's linked to people that get leukemia, for example. Those are the kinds of things that you get out, and it's massively dependent on the existing databases, on what we already know about biology. And so if you look at the overall pipeline, and this is what I've written down in sort of reasonably gory detail in links from this blog post, you generate the sequence. This is what's sort of handed to you as a big file, a couple hundred gigabytes or so. You do this mapping and variant calling, and I did this with the bcbio pipeline, which orchestrates the BWA SAM tools and FreeBayes tools, which are all open source tools that go from this raw data to your set of variants. And then I used VEP, which is a variant annotation approach, and Snipedia to call things using the Gemini, orchestrated by the Gemini pipeline. And again, I'll talk a little bit more about these in a minute. So, OK, so this is sort of the level of abstraction. Before this is sort of the level of abstraction I was used to operating at. I don't actually work on human genetics or genomics. I do a lot of programming. I do a lot of stuff non-humans, but I don't work with humans. So I needed to, for the purposes of this talk, I needed a test data set. And I didn't clearly get permission to sequence my own children from my partner. So, and I didn't really have the money either. So I went with this set of public sequences from the Personal Genome Project. And these are sequences from a trio, a son, father, and mother of the Ashkenazim. These are, a population of Jews that have a particular genetic structure, and I'll talk about that in a sec. So the reason you're interested in a trio is, if you remember, there's two sources of genetic variation. One is the recombination between the parents' chromosomes, and then the novel mutations that come in just from the process of moving the generation forward. And if you do a trio, you can very clearly distinguish between differences or variants that are novel in the son that weren't in the father or the mother, and things that were inherited from the father or the mother, or clearly within the limits of the data that you have. And so a trio is sort of your baseline if you're trying to understand what's hereditary and what's de novo. That's sort of your baseline kind of data that you need. So one of the things that I both like and dislike about human genomics is that you run across, fairly frequently in the literature, you run across some pretty horrifying statements. So this one is, the Ashkenazi branched off from other Jews about 2,500 years ago, during right around the destruction of the first temple. They flourished during the Roman Empire, then, quote, went through a severe bottleneck as they dispersed, reducing a population of several million to just 400 families who left northern Italy around the year 1000. I don't know what happened, but a severe bottleneck is not a good thing, right? We're pretty clear on that. Several million down to 400 families meant a lot of them died. This genetic bottleneck is also one of the reasons why the Ashkenazi Jews are such a great, this is always one of these horrible things to say, right? They're a good target for investigation of inheritance because they went through a bottleneck where there was relatively little genetic diversity 1,000 years ago. And this means that we can really understand what happened in terms of population structure over the intervening millennium. Okay, you go out, you download a 110 gigabyte file that contains all those sequences and the quality scores. This is a BAM file. You download the human genome, which is about 3 gigabytes or so. Generally, this human genome is represented in haploid form, which is, say, one of the two chromosome pairs is what's represented. You also download lots of databases of varying size. And again, full instructions for actually running all of this stuff are available here. But it takes too long for me to do much interactively. the first thing you need to do, as I said, is mapping, and you can use software like BWA, and what you do is you give BWA a set of this data and a reference genome. BWA is written in C or C++, I'm not actually sure. It's open source if you're interested. And it yields tab delimit by a guy named Hung Lee. And you give it these two things, and it gives you tab delimited output that basically say this read of this name, this sequence of this name, maps to this position on this chromosome, this position on this chromosome. Here's, I elided the DNA sequence and the quality scores, and here's the alignment. And you get approximately 2 billion of these entries, and it's tab delimited, it's text format. And this is what contains information about where each of the reads maps, and how well it maps, and what differences there might be. Then when you do that, you get out this kind of output, which tells you how frequently you've observed each position in the genome that you're sequencing. So here you can see there's a histogram, it's depth of sequencing coverage. It doesn't show up that well from here. On average, every position in the reference genome was touched 50 times or more by this digitization. Some were sequenced much less. These are probably half haplotype situations where there was a big difference between one of the parents and the other parent, so they only sequenced half. And then there's a bunch of stuff that's actually observed. Wow, that arrow did not go the right place. There's a bunch of stuff that's actually observed 0 times. And that's because the current sequencing technologies are sort of approximate and don't always touch every part of the genome. So you get into the sort of weird and annoying data science problem where you really have to account for a lot of noise in the data. FreeBayes calls the variance, and what FreeBayes does is it takes in this set of aligned reads. So you have the reference here, you have all of the reads that are aligned, and it counts the number of sequences that are actually aligned at any given position and calls the variance. So you can't actually, wow, that didn't show up terribly well. You can actually see where, there's a 9 T's and 9 G's here versus 8 C's and 8 A's here. So probably this is a half and half situation where the mom had a C and an A here, and the dad had a T and a G here, or vice versa. And that's what we're measuring with this FreeBayes variant calling approach. And so at the end, you get out a list of places where these reads differ from the source genome. And it turns out that most of these differences are completely uninterpretable. So just to give you an example from this Ashkenazi Sun genome data set, there are about 5.5 million variants. So in a 6 billion base pair genome, there's about 6 million places where the Sun differs from the reference. So that's about a 1.1%, right? 1 in 1,000. Between genes, there's half of these are in between genes. We have no idea, in general, we have no idea what they could affect, if anything. They might very well be neutral. We have a bunch that are within genes, but in between the bits of the genes that we think matter most. And so overall, about 5 million of these 5.5 million total variants are things that we know fall in areas where they're very difficult to interpret. So you get about 500,000 remaining, and this is all in the context of that 2%, only about 2% of the human genomes makes genes, and maybe 5% of the genome is thought to be functional. So there's all of these variants, most of these variants are neutral and fall in regions that just don't seem to matter. And even when you know what they matter, the green here is what's called about 50% of the data is likely to have no particular effect on the gene. So there's a massive amount of accumulation of neutral variants, neutral mutations in the genome over time. It's both, they give you good markers, but they also, for things that might be around them, but they don't really, it's like, imagine that somebody gave you the diff of a really large software package against an older version and said, look, there's 1% of the characters have changed between these two programs. Say, well, how many of those changes matter? And be like, well, we can ascribe specific function to 1% of the changes or 5% of the changes. And they say, well, what about the other 95%? I have no idea. So it's an interesting software engineering problem in effect. Okay, you've got your variants. Let's suppose that you've got your list of 5.5 million variants, and you want to know what to do with them. This is where this great blog post, How to Use 23andMe Irresponsibly, comes up. And so I don't know how many of you know about this. 23andMe was a service that would sequence a subset of your genome, essentially observe a subset of your genome and give you a medical report saying, you have this likelihood, based on what we know about how the genome works, you have this likelihood of this kind of medical problem and this likelihood of this kind of medical problem. And the FDA stepped in and said, look, like, you can't do this because we just don't know that much, and so you're really counseling people based around high uncertainty statements. And it's an interesting push me, pull you between sort of the Silicon Valley regulation is always bad and the federal government saying sometime we're going to regulate you anyway. Anyway, so there's this great blog post like, well, you're not allowed to, 23andMe is not allowed to give you these disease calls, but you can go to SNPpedia and you can actually sequence your own genome and go to SNPpedia and do your own analysis. So why not, right? So I decided to set out and look for something that was highly abundant in these Ashkenazi Jew populations. And I found that the son was a carrier for what's called Canavan disease. And there's a particular variant, RS129-48217, which is in a gene that I think we still don't know the function for with in great detail. But basically, we know that if there's an A in this position, you have the risk of Canavan disease. And if you have two A's there in both of your chromosomes, in both of your pairs of chromosomes at that point, you're likely to have the disease. Otherwise, you're a carrier. So we went looking, and this is actually what the raw genetic sequence looks like, the reads. And you can see that about half of the reads, half of the sequences here, have a T at this position. What this tells you is that the son, and actually I went and looked at both parents, they're all heterozygous for this. One of their two chromosome pairs has the allele for Canavan's disease. So approximately 1/4 of their children would have the homozygous allele and probably be affected by Canavan disease. Now in this case, I was pretty sure that the son didn't have it already because children that have it rarely live past age 10, and the son is I believe in the 30s based on the medical records. So pretty unlikely to have it, but it's an interesting example of, it's just nice to go in and look at the genome sequence and see this. Now one interesting thing, just to sort of highlight the annoyance of genomics, I said an A allele here, or SNPpedia says an A allele here at this position, and I showed you a bunch of T's. What's going on? Well, it turns out the variant in the genome sequence that I called is actually a T. SNPpedia says A is the problematic variant, but that's on one particular version of the reference genome. On an older version of the reference genome, which is actually what we called variants on, the relevant gene is on the reverse strand, so DNA is actually double-stranded, and I didn't really want to introduce this complication, but there's actually, for every one of these bases up here, there's a complementary character, CT, CC, A, G, A, T, and what really is happening is the gene is going in this direction, so this is an A in the reference frame of the genome of the gene that we used. So this is just another example of sort of version or dependency hell. There's a lot of links between sort of how the genome works and, yeah, do you have a question? I don't know, actually. Email me and I'll put that in the blog post. Okay, so another fun thing you can do with all of this is you can look at things like human migrations. And you can ask, does what we know historically about where humans came from and how humans migrated, does that correlate with genetic distance? And once you have that list of variants, you can actually start to construct distance metrics between different populations and say, you know, how does this work? And it's actually a fairly straightforward connection once you have, say, a couple hundred or a couple thousand genomes with these variants called. So I have to read this. So this is one of these super cool maps that the map shows the initial colonization of Europe approximately 40,000 years ago from the Middle East. That's the blue arrows. The red arrows are, the green arrows are the retreat of these populations into 4 major groups based on the glaciation, and then the further expansion. And you really can't see them in this, but the yellow arrows are the migration of the barbarians into the Roman Empire, where they did what barbarians do. And what's super cool, and this is a really interesting paper to read, is that you can actually do an ordination or a distance plot, a distance metric, and show that the genetic similarities overlaid on a map of Europe almost exactly correspond to the geography of Europe. So the mutation rate is fast enough that as these populations migrated, and I'm not sure what the right word is, mated within, they constructed their own set of isolated genetic variants that without migration led to sort of a local population structure that can be overlaid directly on the geographical map. And so this is the kind of thing that we're starting to construct really for everybody, every population on the planet. Another thing that you can do, and this is alternately people get either really excited about this or really down on this, is you can say, well, suppose I take 1,000 people with, say, coronary artery disease, and I take 1,000 people from similar backgrounds that don't have coronary artery disease, and I ask, are there variants that tend to be associated with a group of people that have coronary artery disease that aren't in the other population? A fairly straightforward data mining task. And you can get these plots that show, these Manhattan plots that are called because you're looking for the skyscrapers, that show you where there are variants that actually correlate with particular diseases. And this is what's known as a genome-wide association study. There's a bunch of problems with them that basically have to boil down to correlations, not causation. Just because you have a big signal here doesn't mean that that's actually what causes coronary artery disease. It's just correlated with it. And there's a bunch of other things. And this, again, points to this. This is one of the things that I find enjoyable about this is really still the data science problem from hell. You have massive amounts of data. You need large sample sizes, complex statistics. The less fun bits are that every database has a different incompatible set of identifiers, which might sound familiar to those of you. Large effect, and then there's a biological problem, which is that large effects are rare. Very rarely do you see something like this that underlies a complex disease. Okay, so I'll finish up with a couple observations. So where next? In the short term, 2 to 5 years. In the medium term, 10 years. And in the long term, there's some interesting things happening. One is in the short term, just going to be lots more data. So there's going to be millions to billions of human genomes coming. There was an estimate that about 300,000 human genomes were sequenced in 2014, mainly in research labs. Tumor and somatic data, meaning from specific diseases that might be caused by mutations or errors in replication. Time course data, there's this great post down here at Nature about the narcissome, where a guy named Mike Snyder, who's out at Stanford, sequenced himself and his blood and everything on a recurring basis for, I believe, months, and actually caught his transition to type 2 diabetes in sort of the blood level, with sort of the genetic underpinnings at the blood expression level. And then we're also getting newer sequencing data types. These are all things that are definitely going to happen, that are already happening in the next couple of years. The short-term software problems are interesting. There's a lot of open source Python projects now. So bcbio and Gemini are how I produced some of the results earlier. Integration between tools is a poor point because we don't, is a weak point because we don't get a lot of support in science for working with other people, somewhat surprisingly. I would just say that the optimization of specific approaches isn't so important. What I'm mostly interested in are these sort of flexible and robust libraries that would let you construct your own queries in various ways. And I'm happy to talk more about that. General speed and there's technical problems are issues, but they're being worked on actively in the field. It gets a lot more interesting in the medium term, though. We're going to be sequencing everything all the time, and we'll still have no idea what any of it means. And this basically boils down to data integration and data mining problems of the data types that I've shown you. Large-scale sequencing is also being extended to ag, ecology, and veterinary medicine. And so I don't know if I said this at the beginning. I'm now in the School of Veterinary Medicine at UC Davis, and I'm not by any means a vet. And the reason is this data mining problem. They know they're going to be sequencing. You're going to bring your dog in, and they're going to sequence your dog to find out what actual breed it is and what genetic disorders it might be susceptible to. And we're still developing the infrastructure to do this for humans, much less for animals. And then if you want the totally scary medium term, we're going to be able to edit genomes. We already can edit genomes fairly easily, but we don't know what to edit. Imagine that I handed you that Perl 8 bolus of source code and I said, fix it. Okay? So here's a text editor. Here's a lab that will take that Perl 8 genome and build a human from it. Now fix it.
It's illegal to compile.
And it's illegal. Well, you don't compile Perl 8 anyway. It sort of manifests. But you know, you can run it. So if you want to be totally scared, read up on Gene Drive, which is a way of forcing genetic editing to spread throughout a population. And think about that a little bit. Okay. Longer term, here's the really fun bit for me. I expect, barring horrible accidents, to still be around in another 15 years. We've only had large-scale sequencing in the human genome for 15 years. Now free associate, you know, dollar-level sequencing of the human genome, quantified self, and the Internet of Things. And nobody has any idea what's going to be happening. And I think this is a really tremendously exciting time to be in biology. If you're interested in any of the stuff that I've talked about, there's a lot of open source stuff. Genomics is really large and largely invisible. It really has its own culture, so you should come work for me for free, and I'll help introduce you to all of this. I'm just mostly kidding. But there are some specific ways to get involved. The bcbio and Gemini projects are very standard open source projects, and they're on GitHub. They have pull requests. They have a pull request system. And they're interested in things that I think some people in this audience would be able to help with and be a great way to start getting involved in some of these issues. I'll leave this slide for later. Just say if someone tells you they're going to sequence your genome and they're charging you less than $1,000 today, they're not actually sequencing your genome. And the way where you should look is personal genomes. And with that, I'd like to thank you all for coming and take any questions. Thanks. So one quick note is just that to actually do all the variant calling and so on, the instructions are on the blog, but they take a couple weeks of computing. So you know, I didn't want to talk too much about it. It's kind of boring technical stuff. It's awesome technical stuff, but it's like not talk-worthy.
Great. So we have a few minutes for questions. If you have a question, please come up to one of the microphones.
Hi, thank you for the nice talk. I would like to know if you have sequenced your genome.
I have not sequenced my genome, no. I'm actually relatively poor because I'm in academia. So you should totally come be an wait. Okay, well, anyway.
Do the existing open source tools, do they already do things like hardware acceleration, or is it just kind of running on a CPU?
Very interesting question. So the question is about hardware acceleration, and the answer is there are companies that sell hardware accelerated versions of the various tools. And the one problem is that the scale of the problem is such that very few people can afford the dedicated hardware. A lot more people are generating data than can afford the upfront investment in dedicated hardware. And the speed increases are not tremendous. They're only about 100 to 1,000-fold, or sorry, 10 to 100-fold, which actually isn't enough to justify the cost always. So,
thank you.
It might be too long an explanation, but would it be possible to explain how the genome is actually digitized? And well, either way, could you give me a kind of a ballpark figure for like how much of the genome that you get back is accurate, or like what the error rate actually is?
That's a really good question. You get back somewhere around 95% of the genome, and I think it's in the neighborhood of 98 to 99% accurate or greater. The coolest new technology, which is much easier to explain, is you actually have a pore, you have a membrane, and you watch the DNA going through the pores and observe changes in electrical current as different bases go through each pore. That's called nanopore sequencing. It's not yet feasible for this scale, but it will be in 2 or 3 years, most likely.
Awesome, thanks.
Sure.
Can you speak to how you know that a given variant belongs at a given place in the reference genome? Like, how do you know it's not just different? Like, what's the likelihood of getting a thing that appears to match but actually belongs in a different place in the original genome? So
it's a really good question. The short answer is if the genome were random, which we would have a different audience here if the genome were random, you would have essentially 100% likelihood of knowing the exact location. As it is, about 50% of the genome is repetitive, and that causes exactly the problems that you're talking about. And I think that the way currently of dealing with that is to rule out as many things as possible and not call in ambiguous situations. But it's still a very tough problem, absolutely. The challenge is always, how do you do accurate calling in the face of a lot of noise and do it quickly? Right. And there's always these trade-offs, and that's, yeah, the tricky bit.
I had an impression that 23andMe does sequencing of all major SNPs. Why would you need more than just major SNPs?
You know, I don't have a short answer for that. I should probably. The general answer is that if you're looking for heredity, the major SNPs are enough to tell you who your parents were and who your grandparents were and where you came from. If you're looking for rare diseases, then they're not going to be sufficient, because by definition, they won't be they won't be major SNPs. They won't be major variants. So that's sort of the short answer.
we talked earlier. I have a plug and a question.
Oh, excellent.
So I'm doing an open space on genomics. It's actually the similarities between genetic code and computer code. So it'll be fun for both audiences, and that's tomorrow at 4:30.
Tomorrow at 4:30 in open space.
And
excellent.
I'll be posting it on the open space. So the question that's really been pressing on my mind is we were talking big about the cheap sequencing. It seems like things are dwindling a bit. Do you think the $100 genome price point is feasible at this point, or are we seeing diminishing returns?
I guess I sort of have two answers. One is we will see $100. I mean, in the latest, the nanopore sequencing technology is basically a USB-sized chip that you just put DNA on and it sequences it. Right now, it's pretty low throughput, but wait 2 years, we're good. We also think Illumina right now doesn't have much competition, so they're probably not releasing their latest technology until somebody else does, and then bang.
Okay. I was just trying to see if it was similar to the promises of getting 12 gigahertz processors some days. You know, there's physical limits.
I guess the other point I would make is I'm not sure we need more data. We can already generate plenty of data. We don't know how to interpret it. So.
Very true. Thanks.
Hi. So you seem to have found a great position. Is that better?
Oh, cool.
You seem to have found an awesome position in academia that values and rewards computer stuff. That's awesome. Are there any? I've heard that that's not always true, and that academia and tech industry are not always like.
So my lab is laughing back there.
Yeah.
This has been some version of an insane gladiatorial fight for the last 6 years, and I actually have a whole talk on this that I gave last Friday on how to get tenure while being an open scientist, and it's not easy, and we're not. I don't think data scientists are currently rewarded in academia. And it's a real shame because it's an utter disaster for biology. And if you had a more specific question, sorry, I just was like, I just had to laugh.
I was just asking if there are, I guess, places that are doing this really well, or you're like the lone person doing this.
You know, human genetics and genomics is bigger. The problem is, so Jake Vanderplas, who will be speaking here after the break, has this great blog post or great pair blog posts on the big data brain drain, right? So suppose I tried to sell you on two positions. One position was spend 5 to 7 years getting paid about $20,000 a year to get a PhD and learn all you need to know to become a biologist. Then spend 3 to 10 years as a postdoc, and then maybe get a tenure track faculty position and spend another 6 years writing grants. Or you can go work with those same set of skills that you'll be able to put to use 20 years in. You can go work in Silicon Valley for a company that will pay you $100,000 a year to do the same thing that will take you 20 years to get to in academia. And I think that more or less lays out the problem.
Now I'm sad. Thank you.
Sorry.
Okay, so we have time for one more question.
I'm actually curious. I'm coming back to the previous question, not the one just before, but the one previously.
Okay.
So the cost of sequencing is going down more and more, so which means that you just say the nanopore is a USB-side device. So that means that at one point we're going to be able to see individuals actually acquiring sequencing technology, and I'm actually curious if that's a good thing or not. So as a professor, what do you think about having a large public availability on large genome sequencing?
So if everybody's genome everywhere is known, is that a bad thing?
Everybody's genome, or the technology to actually sequence the genome being made publicly available.
I mean, I guess I would go back to the internet. Say there's a lot of many great things about the internet, and it also happens to let the NSA read everything. So you know, I guess I would. This is probably a really stupid response, and there are people that study this full time that'll give a much more nuanced response. But every technology has both good and bad sides, and I think for human genome sequencing and editing, the upside for biomedicine and longevity are just so tremendous that we have no choice but to go forward. I don't know what horrible things will happen.
Have you seen the movie Gattaca?
I actually haven't. I really should.
You really should. Thank you.
Okay. Thank you.